So let
Hello.
me introduce you, and then we can get on with your presentation.
Sure.
Dr. Mohsen Akbari is an associate professor of mechanical engineering at the University of Victoria. In addition to his professorship, he is also a member of the Center for Advanced Materials and Related Technologies, Center for Biomedical Research, and the co-founder and CSO at 4M Biotech. Dr. Akbari obtained his PhD from Simon Fraser University and his postdoctoral training at Harvard Medical School and Brigham and Women's Hospital. He is the recipient of several awards, including the Michael Smith Foundation Idea to Commercialization Award, the NSCRC Postdoctoral Fellowship Award, BC Innovation Council Research Award, Kaiser Foundation Award, and recognized as the Canadian Rising Star in Global Health by Grand Challenges Canada. Dr. Akbari's research lies at the interface of cellular biology, biomaterials, and microtechnologies and aims to develop innovative approaches for disease modeling and drug delivery. He currently holds funding from CIHR, NSERC, CFI, the Michael Smith Foundation for Health Research, the BC Cancer Foundation, and the Department of National Defense. He's a guest editor of Micro Machines and a member of the board of directors of the Canadian Biomaterials Society. His research findings have been published in 90 peer-reviewed journal papers and book chapters with an H-index of 32 and over 4,500 citations. His work has been featured on the cover of 10 journals and reported in various media outlets such as CBC News, Time Colonist, BBC News, Google News, Science Daily, The Telegraph, and Fortune. And today he's going to talk about glioblastoma-on-chip model for preclinical drug screening.
Thank you so much, Dr. Abano. And I do want to add he's one of our more anticipated speakers in the lineup today. He is well connected with our group, with Sherry Monroe and our staff. So again, I want to thank him before.
Sure. Can everyone see my screen?
Yes.
Can you see my slide? Okay, that's great. Thank you very much for your kind introduction. I'm very excited and pleased to be here on behalf of my team at the Laboratory for Innovations in Microengineering to talk about our efforts on using microfluidics and biomaterials in developing a cancer-on-a-chip model for preclinical drug screening. So the focus of my talk today will be on glioblastoma. However, the concept, we are using the same concept for other types of cancers. Before I start, for those of you who do not know where University of Victoria is, we are in Canada. We are in Canada's southwestern coast in a little island called Vancouver Island. Vancouver Island and Victoria, they are known for their beautiful gardens, the Butchart Garden. If you ever visit Victoria, feel free to shoot me an email, and I'll be more than happy to take you to the Butchart Garden, which I love, and I have visited more than 20 times over the past 5 years. We also have amazing hiking trails for those of you who are interested in hiking. And of course, we are the capital of BC, and we make all the rules for the rest of the province. So my lab started in 2015 after I finished my postdoctoral fellow at Harvard Medical School. And since then, we have formed a relatively large group of researchers and trainees and with different backgrounds, including postdocs, PhD students, and undergraduate students. This is my team. You see a little bit of variation in the height, but I'll make sure that the average height is my height because I'm the PI and everything should be centered around me. So we lost 2 tall members however, we are looking for that reason. We are looking for 2 new students who are as tall as they are. So the topic that I'm going to talk about today is cancer. So cancer is the second cause of death globally. Unfortunately, in Canada, that's the first cause of death. And every year, 10 million people die from cancer. So cancer is basically a particularly common cause of death in developed countries such as Canada, where people are less likely to die due to infectious diseases and other types of causes of death in low-income countries. Among different cancers, my lab has been interested in glioblastoma. Glioblastoma is a deadly type of cancer with an average survival time of between 12 to 18 months, and with only 5% of patients survive more than 5 years. Glioblastoma is the most common and aggressive form of primary brain tumors in adults. So it originated in the brain, and then it spreads in the brain following a finger-like pattern, which means that the tumor has a tumor mass. We have a tumor mass, and then also we have a couple of cancer cells infiltrating a few centimeters within the tumor. And then you will see in the next slide that this feature is one of the reasons that the current therapies do not work. Another feature of glioblastoma is that it's different from patient to patient, and then also the tumor is evolving. The genomic landscape of the tumor is evolving over time in a single patient, which makes it very difficult to treat. So management of glioblastoma is done by first doing a surgery. Surgery is the first step of treating glioblastoma, in which a tumor mass is removed to the extent that is possible. Keep in mind that surgeons are dealing with a delicate tissue, a delicate organ in the body. It's very soft, and it's the center of nervous system which controls our emotion and then also our activities. So they have to be very careful when they're removing the tumor. And then, of course, during the surgery, they remove the mass as much as possible, and then collect biopsy samples for further studies. Complete removal of this tumor is almost impossible due to the diffusive nature of glioblastoma. As I mentioned, some of the cells infiltrate a few centimeters within the brain tissue. Therefore, it's not possible to remove those cells surgically. Also, it's very difficult to tell the difference between the edge of the tumor and then the main and then the normal brain tissue. So this means that after the surgery, some part of the tumor will be left behind, and then other treatments should be performed. So chemotherapy is an effective treatment similar to other types of cancers. So chemotherapy after surgery is a routine way of treating glioblastoma. The choice of drug therapies for glioblastoma is unfortunately very limited. There are only 4 FDA-approved drugs available in the market, and temozolomide, which is an alkaline agent that can cross the blood-brain barrier, is the most common type of therapeutic that is being used at the moment by the patients for treating this disease. And then also studies have shown that when chemotherapy is combined with radiotherapy, then the survival rate can be increased to up to 2 months. But still, the maximum survival rate is between the range of survival rate is between 12 to 15 months. So there are a few challenges in terms of treating glioblastoma. It's a very hard to treat disease. One challenge is chemoresistance. Because of the diffusion barriers of the tissues in the brain, and also the presence of blood-brain barrier, the lethal doses of the chemotherapy does not reach a few centimeters deep into the brain, and therefore the cells that are deeper into the brain, they receive lower doses of chemotherapy, so they don't die, but they become resistant to the drug. Also, tumor heterogeneity and patient-to-patient variation is another challenge, which means that that one drug may, or one treatment regimen may not work on the on all the patients. So and then also, of course, there is the issue of the blood-brain barrier that prevents using many available drugs that are effective for other cancers in the brain tissue. So we need tools to evaluate the safety drugs before they can be used. There's a need for developing new drugs, and then for that reason, we need to take a look at the drug development process. I'm sure many of you are familiar with this process. It starts with testing thousands of compounds in vitro at the discovery phase. Then a few hundred selected compounds will go into animal studies, and then a handful of compounds will be selected for clinical trial until one drug enters the FDA process, approved by the FDA. It's a lengthy process. I'm sure you know that. It's a very expensive process. And then in the context of glioblastoma, it has several challenges. The challenges with using current in vitro drug discovery systems is that the majority of in vitro studies rely on two-dimensional monolayers or traditional cell culture systems, which do not mimic the complex three-dimensional environment that the cells experience in the brain tissue. Also, these 2D monolayers, they do not model the diffusion of the drug within the brain tissue. As you, if you remember, the drug, there are cells diffused a few centimeters deep into the brain, and then for us to be able to properly mimic that diffusion, or that the gradient of the drug diffusion in the brain tissue, we need to be able to create an environment that actually models that diffusion, and two-dimensional monolayers cannot do that. Also, there's a challenge of blood-brain barriers and modeling the blood-brain barrier, and in a way that it mimics the physiology of the blood-brain barrier, and the selective, the ability of the blood-brain barrier to selectively transport drugs. And also, there is the issue of organ-organ interaction, which is not possible to model in the two-dimensional monolayers. In vivo models are more complex environments in terms of modeling the actual complexities of the tissue, and then also organ-organ interactions. However, in vivo models, especially at the drug discovery phase, can be quite expensive. We have issues with interspecies variability. There are ethical issues, and then there is a movement towards reducing the number of animals, the 3R movement, and then also they are low throughput. Although there are some medium throughput methods available, however, they cannot be used for testing hundreds of thousands of compounds. So there's an unmet need to develop biomimetic ex vivo models, human-based ex vivo models, to understand the tumor formation, to develop new drugs and progression, and then also to test the safety and performance of existing or new therapeutics in preclinical settings. So my group, along with other groups around the world, we believe that microfluidics, which is the science of using small-scale channels to manipulate small amounts of fluids, can help with developing these or with addressing this current unmet challenge. So in microfluidics, we are using conduits that have the same size as your hair, hundreds of micrometers, and then we deal with small amounts of fluids in the order of nanoliters. So the benefit of microfluidics is that we can control the physical and chemical environment surrounding the cells and tumors at the cellular level. because we reduce the size and we use the small size channels and cell culture microwells, we can increase the throughput at a very small footprints. The process of manufacturing microfluidic devices is low, and then also we can incorporate many valves within these microfluidic devices, which helps us with improving the automation of the system. But one of the main features of microfluidic which can be beneficial for the drug discovery and process and drug screening process is the small scale that enables us to use small amounts of reagents, expensive reagents, and then a small number of cells, which helps us to reduce the cost of treating, of developing basically different drugs and screening drugs. So this is a slide that I usually show to my students in my microfluidic course. I usually tell them that if you do a quick and dirty back of the envelope calculation, but assuming that there is only only the costs of reagents and the cells, and compare the performing a drug screening in a 96 well plate with a miniaturized well plate system, which can be developed using microfluidic system, you will see that for testing 10,000 compounds, different compounds in triplicates, which I'm sure is more replicates are being used by industry. And if you use 50 microliter at the cost of $1 per microliter, the reagent cost for testing 10,000 different compounds would be something around $1.5 million. Again, these are very rough numbers, and that may not be very accurate. And also a cost of cells, assuming that we are just using cell lines, not even patient-derived cells, would be something around $900,000. And the total cost of testing 10,000 drugs in a current 96 well-plate platform will be around $2.4 million. And then keep in mind that I neglected the labor cost, cell culture, material, equipment, capital and maintenance, and then molecular assays and other costs that are involved in assessing the quality of the cells. However, if we now reduce the size of these wells to micro wells, and then instead of using 96 well plates with the size of a few millimeters, if you use wells with the dimensions around 100 to 500 micrometers, something like this that we have built in my lab using microfabrication methods, then the reagent cost will drop down to $3,000, and then the cell cost will drop down to $6,000 with the total cost of around $10,000. So you see that there is a few order of magnitude reduction in the reagent and cell cost associated by simply reducing the size of the microwells to the wells to micro scales. So this is basically the benefit of using microfluidics in terms of cost and then savings in the reagent use. However, similar to many biological systems, brain tumor is a very complex environment. So for us to be able to re-engineer the brain tumor, we need to first look at the structure of the tumor, the tumor itself, and then also the tumor microenvironment. So if you look at the brain tumor, we will have a tumor mass which is most probably has a necrotic core due to hypoxia. So we have hypoxic core with some necrotic cells, and then we have a tumor edge which is surrounded by a matrix, which is the brain matrix. In the matrix, we have a combination of different proteins, polysaccharides, and then also a number of cells that are present in the microenvironment of the brain, including microglia cells, astrocytes, and neural cells, and then so on and so forth, and then of course immune cells. And then we have a vascular system. And then this system, if you take a closer look at the tumor microenvironment, we can even see the gradient of molecules that are secreted by the tumor due to the, and then also we'll see the gradient of even the matrix of stiffness, mechanical properties, and porosity that is generated within this complex microenvironment, tumor microenvironment. So it's a very complex environment to model. However, similar to many engineering problems, we simplify the problem, we break it down into smaller components, and then we start from simpler problems, and then we will progressively increase the complexity of the system. So for that, our first focus was on modeling or developing a way to create the tumor mass itself reproducibly. So there are many different ways of doing that. The first method was hanging drop cell culture systems, which is a very simple system. There's a well which has a pore connected to the entrance of the well. So what they do is that you create a cell mixture, a mixture of cancer cells in cell media. You deposit the cell mixture. Because of the capillary force, you will form a little sphere at the tip of these little hole, and then the cells start self-aggregating, and then they'll form this little tumor spheroids. So it was at the beginning was a very, very effective method to create tumor spheroids. However, you can imagine that if you want to make many of these spheroids manually using pipettes, it could be very time consuming, and it could be very low throughput. So there were other methods developed lately using microfluidic systems, and these methods are based on using emulsions and droplet-based microfluidics, in which you create little particles, little particles with cells encapsulating in them in a two-phase flow system. So you have an aqueous phase, which is basically formed in an oil phase. So the process helps us to create many particles in an automated manner. However, the challenge with these systems is that we usually create non-uniform cell numbers within each of these microparticles, and then the cell death is generally very high because after we collect these particles, you need to wash them several times, and then the washing steps can be harsh to the cells and can increase the cell death. And then also to create these microfluidic systems, you need to have access to a clean room facility, which is a high-tech facility, and it's very expensive. So another method is to simply reduce or shrink the current well plate systems, shrink their size, and create micro well arrays. So this system is pretty effective in terms of creating basically microspheres. However, the problem is that when you want to seed these wells at this small scale, you will end up with non-uniform cell seeding. This is a challenge that many of the companies that are commercializing these technologies are facing, and they raised this during our conversation. The process is still time-consuming because when you want to seed cells, let's say in a 6-well plate, then you need to have to be very careful to deposit the cells properly to make sure that at least the number of cells that go into each well is close to each other, and then hopefully you get some level of uniformity in terms of the formation of the spheroids. And then also similar to the previous microfluidic system, you require to have access to a clean room facility to create the molds and replicate them and replicate their features in this basically the material. So what we developed and our solution to this problem was a technology that we call it self-filling microwell array systems. So the technology has two benefits. The first benefit that this technology offers is that instead of using the current clean room workflow to create microfluidics, we rely on using 3D printing technology, in which it allows us to create molds using the biocompatible resins. So the ability of creating using resins and 3D printing to create the molds enables us to create a wide range of different designs, including circular microwells, square microwells, and then also microwells with very different geometries, and I'm not going to get into the details of this, but by controlling the geometry of the microwells, we will be able to control the phenotype of the cells, and then also the size of the spheroids that we could generate in this system. But this is not important at the moment. What is important is that with this method, we do not need to use a clean room facility. We can quickly make the molds. We don't need to go into a clean room and spend 4 or 5 hours to create one design. And then if it breaks, then you need to go back into the clean room and then create it again. It takes less than 10 minutes to create tens of these molds, and it enables us to create non-planar and complex structures, which is a feature that we use to create these self-filling microwell arrays. So these arrays are not planar anymore because we could use 3D printing to create non-planar systems. So the microwell system array is something like this. The size is adjustable, so we can change the size to fit into different designs of microwell, like commercially available well plates, 6 well plate, 24 well plate, and so on and so forth. The unique feature of this system is that when you want to seat them, you do not need to fill the entire well plate with the cell suspension. So we have designed a cell seating chamber which is connected to the wells. So this is the cell seating chamber here. This is the cross section. These are the microwells connected to the cell seating chamber. And this image, you see these microwells are basically around this basically circular well. They are connected to this seating chamber using a guiding channel. So these guiding channels, what they do is that once you seat the cells in the loading chamber, the cells basically roll into these guiding channels, which are inclined by gravity, and then they'll go and then fill into the wells that are connected to the end of these each of these channels. So with that, we were able to achieve very uniform cell distributions in each well, which resulted into obtaining uniform tumor spheroids from different types of cells. We use cell lines, breast cancer cell lines, glioblastoma cell lines, and then also we use patient-derived cells. So we were able to create spheroids with uniform size distribution, and then we also used a one-step rapid seeding approach to achieve this. So just to show that we can use these tumor spheroids for drug toxicity studies, we created 2 types of tumors, cancer cells, breast cancer tumors, and then glioblastoma tumors. And then we treated them with doxorubicin to show that, first of all, we were able to create these tumors, we were able to control the size of the tumor, and then also we were able to treat these different microspheres with drugs, and then quantify the amount of cell death. So what you see here is the tumor on day 1, and then the tumor was treated over 5 days with doxorubicin, and then after 5 days you will see that the tumor the cell death, which is basically the red core that you see here, increases, and then eventually on day 5 the tumor was dissociated, and the cells were basically disintegrated, and then the number of cell deaths increased dramatically. So we quantified this using regular cell live death assays and flow cytometry. One thing that we noticed was that when we compared the drug sensitivity of spheroids and the monolayers, we noticed that because of the diffusion barriers that are present in the tumor spheroids were less sensitive to drugs when we compared them to the monolayers, which clearly shows that using a model that tumor spheroids that model the diffusion barrier against the drug penetration into the tumor can be very helpful in the drug development process. So we successfully created the tumor spheroids. Now we wanted to take another challenge, which is mimicking the tumor microenvironment and then the cells around it. So what we did was that we created a microfluidic device which had a central chamber and two side channels. The central chamber mimics the tumor and then the tumor microenvironment, and these side channels, they mimic the blood vessels that deliver blood and then also nutrients and oxygen and also drugs to the tumor. So in the central chamber, what we did was that we created the tumor spheroids using our self-filling microwell array. We mixed it with a matrix that mimics the matrix of the brain's extracellular matrix. So what we did was that we included proteins such as collagen, fibrin, and then also polysaccharides such as hyaluronic acid. So these proteins and molecules that are present, we know that they are present in the brain extracellular matrix. And then also we adjusted the stiffness of the matrix to mimic the soft matrix of the brain when we were designing this gel. Also, one of the things that we did was that in this phase of the project, we also included neurons, which were basically differentiated from induced human pluripotent stem cells, and then we incorporated them in the gel that was surrounding the tumor. So, in summary, we had a tumor encapsulated in a cell that has in a matrix that has the neural cells, and then also it mimics the proteins and then the matrix, the extracellular matrix of the brain tumor. So the first step for us is to ensure that the matrix is safe to the cells. So we grew the cells, neural cells. They maintain viability, and then also the viabilities were pretty high, very good. Basically, they express the neural specific markers MAP2, 2J1, and we monitored the cell invasion, the tumor invasion in the extracellular matrix. We noticed that the cells that were growing with neurons, they are more invasive, and then they diffuse more into the tissue. We created a chemoresistant model by using temozolomide-resistant cells. And then we noticed that the temozolomide-resistant cells, their morphology and then also their invasion pattern was different. We studied basically the cytokine profiling of the tumors, and then we observed that there are certain cytokines, at least 19 cytokines represented the progressive increase associated with the invasion, angiogenesis, and stemness of the tumors. And then also we performed a proteomic study and showed that when the cells were grown in 2D, they're in 3D in our microfluidic device, their proteomic profile was significantly, in this case, were significantly different than when they were grown in a 2D culture. And then also, even when the cells were resistant and non-resistant, we showed that there is a huge difference and significant difference between their protein levels. So, to summarize, there's an unmet need to develop new preclinical tools that can recapitulate the complexities of tumors and tissues in vitro. We believe that microtechnologies combined with biomaterials and cell biology can potentially offer a solution for this unmet need to create a more realistic drug screening tool in preclinical settings before the drugs are being tested in vivo and in clinical settings. However, more work is needed to validate these new models. We're not still there. We need collaboration with industry and with other researchers to validate these models, specifically by using patient-derived cells. So with that, I would like to thank my collaborator, who is a neurosurgeon, Dr. Brian Toyota, and then also the funding agencies who supported this work. If you have any questions, feel free to email me, visit my website, or follow me on Twitter, and I will be happy to take any questions that you may have.
Thank you again, Dr. Mohsen, for your outstanding presentation. And the floor is now open for any questions anyone might have. Dr. Dabano and anyone from the crowd.
I'll give it a moment, maybe. Does anybody have any questions?
I see one question from Adrian Rubio. He's asking Dr. Mohsen, do you have plans to look at patient tissue-derived tumoroids?
We do. We need to have a new design. That's a very good question. Because when we use these patient-derived tissues, then you will have the extracellular matrix, and then we do not need to design the matrix. But we are working with collaborators on this. It's a little bit difficult to obtain patient-derived tissues at this stage, but we are in the process of getting them.
I have a question. So, fascinating stuff with these microfluidics and being able to, reproduce tumor microenvironments and cultures for screening. Have you looked at ways of opening microfluidic devices to retrieve cells after their separation to do further experiments on them after screening or exposure to a drug has occurred, to be able to do further assays of those groups of cells to which specific drug activity has taken place?
So let me. So the question that you're asking is whether we can open up this microfluidic device and collect the tissues that we're making and then slice them, probably.
Correct.
Yeah. This is a possibility, and then we are working on a new design of the macro well array platform. I have a postdoc who is very smart, and he is working on this technology that combines microfluidic with the microwell array platform, and it enables us to retrieve the tissue and then use them for basically for slicing them for histopathological studies and then other types of studies. At the moment, we are not doing that because the microfluidic device that we're using, it uses a plastic which is basically very difficult to cut, and then the process for bonding is permanent, so we cannot remove the tissue, but take the tissue out. However, the new design is underway, and then hopefully soon you will see those.
And there is one more question from Dr. Steve Miller. He's saying, can you do live cell staining and then use confocal for live dead imaging?
Thanks, Dr. Miller. Yes. Some of the images that you saw in my slides were, especially the neural ones, were confocal images. We can do that. The devices that we are making are completely transparent. We are using a material that is called PDMS, polymethyl disiloxane. Sorry, and then it's transparent. It has been widely used in the microfluidics, and then it has. We can use live imaging. We have done live imaging, and then we can use confocal. We can stain the cells in the chips, and then we can collect the soup. So there are many things we can do with this.
Wonderful. And yeah, he's asking if he can connect with you after the event, Dr. Steve Miller. Awesome. Yeah, so we'll connect you with him, Dr. Miller, via email. And if there's any other questions, you can let us know, or again, you can email and you can connect with Dr. Mohsen. I want to thank you again for your amazing presentation, Dr. Mohsen. And we will be going to our next presentation, which is next generation psychedelics for mental health disorders. So if we can all just transfer over to that room.